So good afternoon. My name is Michèle Silva, and it's a pleasure to have you here for the EAAP Webinar, Poultry Science, Innovations and Future Perspectives. This webinar is organized by the Poultry Working Group, and for those that doesn't know who are we are a group supported by the Pig Commission of the EAAP, and we have a couple of goals that are to support the scientific commissions in EAAP. We also aim to increase the visibility of poultry research while constructing a scientific platform for poultry researchers, and also to collaborate with other organizations and doing networking on the EAAP platform, while offering a channel for cross-disciplinary and intersectoral dialogue. So who we are? So you have me, Michèle Tixier-Boichard, Gurgus Dash, Anastasia de Grande, Annelien Vatijn, Katarzyna Starnicka, and Shelley Trujan. And the key activities from the Poultry Working Group until now, and a couple of the next ones, were the EAAP webinar in 2021 with a thematic Why to Apply Gene Editing in Animals. We were also present on the EAAP Porto in 2022 with a full-day session on challenges in poultry nutrition, health, and welfare. And we have an invited speaker, Dr. Doug Corber from the University of Alberta, and this was supported, this session, by the Canadian Association of Animal Science. And this year in Lyon, we will be co-organizing 2 scientific sessions with other EAAP study commissions, and that will be also supported by the WPSA French branch, but I will talk about this at the end of our webinar. Of course, in every EAAP conference, we support the Pig Commission in the early career scientist sessions, innovations in pig and poultry industry. I have now, before we start with our webinar, some information for the audience. So each presentation is around 10 minutes, followed by, presentation is around 20 minutes, followed by 10 minutes questions. So you can raise your question in the chat, and then we will place the question to the speaker. For your information, so for sure you already have seen that the webinar will be recorded and then will be available on the EAAP website, and after 3 months also available on the EAAP YouTube channel. Don't forget to keep an eye on the EAAP website, because more webinars will pop up in the future, and just check on the web page. I will now give the word to my colleague, Shelley Trujan, and she will be presenting now the speakers and our webinar.
Okay, thank you, Emilia. It's my pleasure to introduce our first speaker, Professor Michèle Tixier-Boichard. She's a senior scientist at INRAE, where she has been working in animal genetics and genomics since 1985, mainly on chicken. She published more than 140 papers in peer-reviewed journals. She chairs the board of the French National Cryobank and coordinates the National Infrastructure of Biological Resource Center for Agriculture Research and its branch for domestic animal, CRB animal, in 2022. She was elected as the president of the World Poultry Science Association. Michèle, we are so glad you agree to come here and open our webinar, and the floor is yours. Thank you.
Thank you, Shelley. Thank you for the invitation and this title, a bit challenging. So I tried to collect ideas that are not usually presented together at the same time. So I will share my screen. Okay, do you see it full screen? Is it okay? So the title I have proposed is in the field of poultry breeding, poultry genetics, of course, but regarding the challenges that are raised by the different dimensions of sustainability. Sustainability is not a new concept, but it is becoming maybe more and more acute, particularly considering, well, pressures such as climate change. So the outline of my presentation will first involve a short reminder of the success of poultry production worldwide. Where does it come, this success? It comes in part from poultry breeding and genetics. Then in our field of genetics, we have the genomics revolution that I will touch very briefly. And then entering into less comfortable areas with the biological limits to selection, the dimension of sustainability and the controversies which arise from looking at these dimensions, and the consequences, what are the new questions for poultry breeding programs. And I will finish on the FAO framework for agroecology, asking where does poultry stand, because you will see it remains an open question, at least for me, and then give a take-home message. So in poultry production, there has been really a continuous growth. I have picked up numbers given by Peter Van Horn at the last World Poultry Congress last year. Over 15 years, the poultry meat production has increased by 65% and egg production by 50%. This is a worldwide market, and the production capacity has been redistributing geographically in the past 10, 20 years. Europe has lost market share, and Asia, mainly China, but also Thailand, and South America, mainly Brazil, have gained market share. So it's really a worldwide dimension. And this trend is continuing because there is a growing demand for animal proteins in developing countries. And also there is no religious ban on poultry, which is actually maybe almost an exception. I said I make it short. So there is a big trend in performance, in production. So what is the contribution of genetics in this trend? So here I have picked up results which are already old, 20 years ago, where it was possible to compare the effects of selection and of the type of diet on the growth rate of male broiler chickens between 1957 and 2001, because 1957 was the generation of the control population, which was kept by the University in USA of Athens. And they kept these chickens as control and compared them with the chickens currently selected in 2001. And they did the same for the diet. So they took, so you see here in blue, the line corresponding to the body weight of the 1957 chickens, and in red, the lines corresponding to the body weight according to age, 21 days, 42 days, 56 days, and so on. In red, the animals from 2001. Then the dotted line corresponds to the diet provided in 1957 in terms of components of energy, proteins. And the full line corresponds to the diet provided in 2001. So it was a very full, complete comparison with the two genotypes received the two diets. And as a reminder, I put here the body weight of the red jungle fowl, the wild ancestor, at 84 days of age, which is below 1 kilo. So you see it's very, very far from also the 1957 chicken, but even more the 2001 chicken. The label rouge, the one we have in France, which is a slow growing label, but it's still a meat type chicken. So it gets to 2 kilo at 84 days because it is the restriction of the label. And then what you can see that the change in diet in the 1957 broilers does not change very much, the growth. And the change in diet increases the growth in modern chicken, but the difference between the modern chicken and the 1957 chicken remains very high, whatever the diet. So there is an interaction between the genotype and the diet, because the diet has a more favorable effect on the faster growing broiler. And there is a significant effect of genotype and a significant effect of the diet, but the effect of the genotype is by far much more important than the effect of the diet. So this shows the great responsibility also of genetics and of the breeding programs in determining the genotypes for the future, because then everything goes from this performance level of highly selected broilers. In layers, the innovation of current poultry breeders, you see here a typical laying curve with the peak. And actually the peak now is almost at the maximum value that you can get, but then you have more phenotypic variation and genetic variation in the second part of the curve when it is slowly decreasing. And in some genotypes, it is decreasing less slowly than in others, up to the fact that now it is considered to increase the duration of lay. The career of a layer, how it was 1 year of lay, now it can be easily 1.5 years of lay, and maybe more. So you get a layer hen which never stops laying almost, which of course is a tremendous trend as compared to local breeds and to the wild ancestor. Of course, this also raises challenges of the good diet for these layers. But this is to describe this very strong impact of breeding on performance. Now, the genomics revolution. So what it is, this is very short in one slide, I admit. So the idea of genomic selection is to make possible a statistical association between performance and molecular markers. Here you have a very simplistic view of the genome. All along the genome, you have AHCGT, the base, and you have variation along this genome. And by genotyping animals and making also individual phenotypes, it's possible to associate or to give a weight, positive or negative weight, to a marker, depending on its statistical effect on the performance. And of course, this can be done classically for production trait, milk production, eggs, but it can also be performed for fertility traits and also for disease resistance, all the more than in this method, you have phenotypes and genotypes on what we call the reference population. The reference population is the population where you get as much phenotypic information as possible. And it can include disease resistance, including challenge tests that is not usually performed on candidates to selection because you want to keep your candidates in good health. They are in a biosafety area, so it's quite difficult to challenge candidates to selection, but you can challenge their relatives and then genotype their relatives in order to make this association between a particular area on the genome and the phenotype of interest. And then when you have confirmed this association on a large number of animals, at least 1,000 animals to make up a reference population, then you can directly select just on molecular markers on the traits, even if you do not measure the traits. Typically on males, you can have a breeding value directly from the genomic markers. For female traits, that is done in dairy cattle since several years now, but it can be done also for cocks on laying traits. It was done in the past, but only on the basis of pedigree relationships and a statistical calculation on association between performance and pedigree. And here also you have a lot of calculation. It's not to make it more simple on the viewpoint of computing, but you can directly select an animal's very young just on its genotypic markers. So since you can make this association on different traits, different regions of the genome, then it is possible to add more traits and to distribute the selection effort on many traits. But always you need individual performance in the reference population to build this association between performance and markers, and you need to update this association regularly. This is a tricky point, depending on the species, the selection intensity, the generation interval, how many times you need to confirm the association. So this is being applied in poultry breeding now, genomic selection, but we are starting to observe, it's not so recent, some limits that I may call biological limits to selection. And this is a very simplistic example about selection on breast meat. So which I have represented very schematically here by non-selected chicken, selected chicken, a highly selected chicken, where you see that the breast meat is becoming bigger and bigger. And we have also a growing incidence of meat defects, of myopathies. At the same time, if the selection is just looking at breast meat percentage, the supporting tissues, bones, or cardiovascular tissues are not selected. If you do not think of it in advance, they do not follow the trend of the quantity of breast muscle. And then we observe fitness problem, which is due to sort of an uncoupling between the tissue, the muscle tissue being produced, and the metabolic supporting tissues, bones or cardiovascular system, which are not improved at the same level. So that's why people started to observe lameness, tibial dyschondroplasia, and ascites. But of course, each time you have a new problem, you look for a solution to solve this new problem. So there are solutions to go beyond the limit by the management, modifying the temperature, housing temperature, modifying the diet, selecting in a new environment, and also mainly proposing new selection criteria, typically on a bone score, and also on oxygen level in blood. So this has been also developed to compensate for the ascites that was the result of of a direct selection on muscle without considering the supply tissue. There is another biological limit. So maybe, yes, you can understand that on this type of limits, it's possible to find ways to overcome the limit, although at some point we can question whether once maybe we don't find a way to compensate for the limit. So this is still open question. But in other circumstances, you have a typical antagonism between functions. And when you have a very fast early growth with excessive appetite, increased fatness, and the adult cannot breed unless they are submitted to a very severe feed restriction. And here you see in 2014, for breeders, you had 90% restriction. So they got 10% to it to become able to reproduce as compared to what they would eat spontaneously. So at some point, we cannot restrict more than 100%, or we still need to give some food to these animals. So it's not so easy to find a way to compensate for this, because it creates a severe welfare issue, because the animals are always feeling hunger. And also, it should be reminded that the genetic correlation between growth and reproduction is not linear. Too small or too large body weight is associated with poor reproduction. So that's why in breeding, you need to check for correlations, of course, but also to the change in correlations over time, particularly when you have a so strong genetic progress in some traits. So now, if we look at sustainability. So what does it mean, sustainability? It has been introduced, this concept, since several years. The idea is to meet 3 objectives, which are complementary and necessary to have a durable, a stable situation or a stable system. So the 3 pillars of sustainability are economy, society, environment. And then the ideal is to balance these three pillars. So what are the positive assets of poultry on this three-dimension system? And what are the challenges to address? So the positive assets. First of all, highly selected chickens are very efficient. Feed efficiency is really good. So this is a very good point from the viewpoint of economy. But of course, they need to be fed a rich diet. The feed efficiency is really optimized because also there is an optimal diet. And chickens are producing relatively low level of greenhouse gas as compared to cattle, of course, but also maybe to the mammals. They don't have the same metabolism. On the viewpoint of society and economy, poultry meat is cheap. So poultry is producing cheap and nutritious food, and it's easy to prepare. So these are the positive assets. But now look at the challenges from the viewpoint of environment. Although chickens have low greenhouse gas emission, they may be quite susceptible to climate change, heat stress, and we now observe a diversification of housing systems up to free-range, fully open-air housing system. So not really housing, since they are open-air. And this is a challenge to address for poultry breeding because the selection environment generally is in closed building for biosafety. And at the level of society, we observe, well, difference among continents, at least in Europe, but also in other continents, you have different cultural values, considering the well-being of an animal, considering also the value of animal food. And we have a growing concern in animal welfare, which is not only being in good health, but also the animal should feel well, which is a great challenge, because how do we know that the animal feels well? So if we continue on these dimensions, so we can observe there are controversies or even sometimes antagonistic relationships if we want to achieve this sustainable development or sustainable production. So regarding feed efficiency, which is a very good point from the economic viewpoint, but it is a rich diet. Chickens very often gets soybean, corn, that can be eaten directly by humans. So we have this feed for animals, food for human debate. And this has to do with environment also, because you know that some forest areas are destroyed in order to grow soybean to feed animals. So all the components of the production system have to be considered. And when we look at feed efficiency, we should not forget, where does the diet come from? Regarding environment purely, high performance animals are known to be, well, quite, to exhibit a very low tolerance to heat. And why it is so? Because they themselves, they have a high metabolic heat. They are producing a lot of metabolic heat because of their high metabolic rate, both the broiler during the fast growing period and also the layer, because it is synthesizing the egg, lipids and proteins every day, which means that these animals now, we have realized in the past years that the comfort temperature of broilers, it's even much less than 20 degrees. It's less than 16 degrees, and layers maybe 18 degrees now. The more, the higher the performance level, the higher the metabolic heat, and then the lowest tolerance to heat. So, which means that as long as the ambient temperature is above 20 degrees, the high performance animals are suffering from heat, and then their performance may decrease, and also their welfare, of course. And then we get to the other risk of antagonism between economy and society. Society considers that free range is good, at least in Europe, because animals express their behavior, they can move, and they have a better way of life, I may say, but I think Werner will explain more to you about this welfare issue. But when they are raised in free range, feed efficiency is lower, and they have a higher environmental impact because they stay longer time on the ground, and they need a higher amount of feed to reach the marketing weight. And on the viewpoint of economy, free-range chickens, because they have a higher production cost, they are sold at a higher price. So it is the consumer's choice. Not all consumers can afford this choice. But at present, at least in Europe, we can see that welfare gets a high priority, high weighing factor in this balance system. But actually, the challenge is to find the balance, because free range, better welfare in the free range system is associated with lower feed efficiency and higher environmental impact. So this is a big controversy, which is not easy to solve at the level of the production system. And then the question is how the breeders act in front of this background. So to remind the basic components of a breeding system in chickens. So you have a pure line nucleus, several lines in general. Breeding and selection is performed in a selection environment at the top of the pyramid with closed pedigreed lines. The size of these lines is confidential information, of course, as well as the number of the lines. But generally, you may have combination of 3 or 4 lines, because from these lines, they are crossed 2 by 2, and then you have a second level of cross to get to the terminal cross. So that's why it is a pyramid, because after the selection, you have multiplication, just multiplication of the pure line, A, B, C, and D, then crossing them. So either 3-way cross, generally when you have a sire line, very highly selected for growth rate, and C and D are the dam lines. You benefit from a crossbreeding effect on fertility here, and also for recombination, because in this animal, CD, they are F1, and at the time of reproduction, you may have recombination mixing alleles of C and D in different order on the chromosomes, which creates a source of genetic variation. And then the CD is finally crossed with A to get the terminal cross. And in this terminal cross, you have new allelic combinations that, of course, did not exist in the parental lines and were not accessible to selection. And for a 4-way cross, it's even more, because you have AB and CD, 2 levels of recombination, well, 2 populations where recombination is taking place, and the final 4-way cross also with new allelic combinations. And these 3 to 4-way crosses are in the production environment, which can be completely different from the selection environment. Now, even more than 20 years ago, you have cages, different types of cages, floor system, different types of floor systems, free range, completely outdoor. You may have suboptimal diet, and you may have also tropical climate, either controlled with air conditioning, which is not so economic and also environmentally friendly, or no air conditioning, and then adaptation to tropical climate becomes needed. So how does this translate into questions for the breeder? At the first level is the breeding goal. For which goal do we breed? Is it always to increase the value of a trait? Is it always more? Or is it to have a target value for a given trait? There are selection methods perfectly well known, which make possible to select for targeted value and for an optimum, and not for always increasing the value of the trait. But if the competing company continues to increase the value of the trait, and you are a sustainable company and you say, well, okay, I will stop at an optimal value, you may lose market share because the competing company may be a little bit better on one trait. And this is all the difficulties. We will come back regarding governance of the chain. So once you have your goal, which trait do you want to measure to meet your goal? And then the big question is how to measure welfare, going beyond longevity and health. Individual data are mandatory. So now we have the help of robotics. This is highly needed for phenotyping behavior, particularly in extensive systems. But for phenotyping well-being, emotions, I don't know. We can use image analysis, but how do we calibrate these images to understand whether the animals is not only well-being, but also feeling well. So these are technological and analytical challenges, not only for genetic system, but for all the poultry production system. Then how to weigh the different traits. Either you have all the traits in the same line, which is quite rare. Generally, specialized lines are derived, and then you specialize them on a subset of traits with complementary goals, so as to combine all these different traits in the final terminal cross. And a big question, which is because can be expensive to perform, which individuals to measure, the pure lines or the crossbred? The crossbred are the one making the production. And in which environments? The selection environment or the production environment? So here, the pure lines are always selected in their selection environment because of biosafety. And the terminal cross, ABCD, are always in the production environment. And because these environments are different, you may have interaction between genotype and environment, and a better growth, for instance, in the selection environment may not lead to a better growth in the production environment, which actually translates into the fact that the genetic correlation between the performance of A in the selection environment and the performance of ABCD in the production environment is well below 1. And this is a challenge for the breeders, so that they need to connect the phenotypes in the production environment with the genotypes in the selected lines. And then we come back to this genomic selection and genomic revolution, because with molecular data, it's quite possible now to connect these haplotypes, these allelic combinations here in the terminal level with the allelic combinations present in the different grandparental pure lines. So this is a good way to go over this problem of different environments, because you may have the marker trait association, you may build it in the production environment. You identify the origin from which lines come the favorable marker alleles or favorable haplotypes, and you breed the pure lines for these alleles, which means that maybe the pure line may not exhibit a better performance in selection environment. But what is your goal? Is to have a pure line with very good performance only for yourself, or you want the final cross to have the very good performance in the production environment? So sometimes you need to rely a lot on your calculation, marker data, and phenotypic data here to be sure that you are making the right choice at the top of the pyramid. And then another types of questions arise is about the consequences of which action can be taken and what are the economic consequences. So genomic selection typically is ranking individuals within lines, but it needs the genotyping, and there is a cost for genotyping. So there is a big issue to optimize the cost of genotyping strategy in order to decrease the number of markers to the minimum needed to decrease the cost. And then thanks to methods of imputation, you can, when you have sufficient data accumulated on the same population, you can infer from the low density markers all the other variants, DNA variants of the genome. And then this, once you have accumulated enough data to make this imputation within a given line, you can decrease the cost of genotyping and still have a good prediction of the genomic breeding value. Then after ranking individuals, the choice takes place at the level of the mating plan, which is an important degree of freedom for the breeder. And there are different methods, models proposed also to optimize mating plan. But it's also possible in poultry breeding to optimize line crossing. and maybe to increase the choice of genotypes and the number of courses. Maybe you can have more than 4 lines and multiply the number of courses you can make to propose a larger choice of genotypes to the producers. And also, as we already discussed, investing in new phenotyping is needed with robotics. Only a few breeding companies can afford this, but as I say, anyway, there are already a very few breeding companies. So they are investing, all of them, in a phenotyping device, such as, for instance, electronic nest, in order to know which hen has laid which egg at which hour in the day, when all the hens are on the floor. Then, because of this investment, also development of methods, there is an increased cost, even if it is under control. It can contribute to increase the cost of the day-old chick. And then there is the pressure from the producers to limit the cost. For instance, for a broiler, generally, the day-old chick should not represent more than 20% of the cost of the final broiler. So that is the uncomfortable zone for the breeders, because they have to adjust their cost, although they have to innovate in technologies, but they have to meet the market demand, which is putting a constraint on the cost of the day-old chick. So now if we go to the enlargement of the vision, taking the vision of the FAO of sustainability of agriculture and livestock production, and in the framework of agroecology. So agroecology, I think it is a topic quite well discussed generally in EAAP meetings, so I don't need to explain it again. But I would like to mention here the 10 elements used by FAO to define agroecology. First of all, agroecology is a system approach, and we can only discuss a contribution of genetics to this. It cannot be only an issue for genetics, but every discipline or every actor in the production chain has to contribute. So here I have proposed 4 elements to which genetics and breeding can contribute. Of course, the diversity, the efficiency, the resilience, and the responsible governance. This is open. Maybe you can identify other elements where genetics can contribute. So diversity. Here I have summarized the heterozygosity of SNP markers that was published in 2019 with commercial white egg layers here, local breeds in Europe, commercial brown egg layers here, the red jungle fowl here, and the broiler, and then African village chickens and South America local chickens. So what you can see that in terms of local chickens, you can find almost any situation depending on the continent of very low heterozygosity local breeds or very high heterozygosity local populations. But except for the white egg layers, the Leghorn, which is always at the bottom of the scale, the brown egg still exhibits some variability, and surprisingly, the broiler lines still exhibit a quite high level of heterozygosity in their genome, in spite their very strong selection since almost 50 generations now. So it may seem as a paradox, but probably the explanation is that the base population, the founders of the broiler selected lines, is quite large. So about efficiency and resilience, this is also a vocabulary I think very often discussed in EAAP conferences. So efficiency for poultry breeding, feed efficiency, what we mean is very frequent, usual trait, unavoidable trait with a great genetic progress. But in which environment, which diet, that what I discussed previously, there is really a dependency on good quality diets and on the associated environmental impacts. And this is a point I think that should be better studied or more deeply because we feel that there are some connections, but they are not well formalized or quantified. So efficiency, it's measured at the individual level. Resilience is a wider concept, more at the system level, In short, it describes the ability to recover from a disturbance, and the disturbance may be from environmental origin. And it's generally more studied at the group level. And a group level can exhibit a more stable performance because, for instance, there are complementary capabilities between individuals in this group. In other words, a group can be stable. It doesn't mean that the individuals in the group are all stable the same, but they may not react in the same way to different stress or disturbances, so that at the end of it, the group can remain rather stable. And here there is an expected positive effect of crossbreeding, which has always been observed, at least in poultry breeding, by classical geneticists and first breeders. I heard in conferences already 30 years ago. That is almost the miracle of crossbreeding. You cross the lines and then the crossbred animals are almost resistant to everything. But this is not always not always verified, but not always well explained also. It's sure that crossbreeding effect has a very positive consequence on resilience. But at the individual level, there is probably genetic variability for adaptation. It's not so easy to analyze, but what we can say for sure, there is not a one-for-all genotype. You don't have an individual which will perform perfectly well in any environmental condition. That's why the job of the breeders is still to propose a choice of genotypes. So now the last point about the sustainability is the governance. Although the poultry breeding, as you could see, is at the top of the chain of this pyramid, but then the market demand is driving the breeding goals. And actually, there is no solidarity along the chain, because the slaughterhouse may fix a price, and then in a retrospect, all the actors of the chain has to follow. The breeder is not giving the tone. It is providing the chickens, but not deciding everything. And the problem that there is no consultation, no solidarity from the top of the pyramid to the bottom at the production level. And this is a difficulty to define an agroecological system in poultry production, because the system in itself involves many actors that very often do not cooperate together. So FAO has developed the tool for agroecology performance evaluation, TAPE, which aims at measuring the multidimensional performance of agroecological systems with an approach at the household or farm level until the territorial scale. And this, at the moment, is still very difficult to apply to poultry systems. Not easy to fit to a system which is industrial and intensive as poultry production is. And sometimes some theory of agroecology conclude that a reconception is needed, but you still need to meet the market demand when you are a breeder. So that's why this is still an open question, not so easy to solve. And I think at least a better solidarity along the chain would be quite needed to improve the sustainability of the system. So the take-home message is that poultry breeding has major assets in hands. So then I'm happy to take critics, because of course I am a bit biased since I am a geneticist. I still find that we have a lot of interesting topics to study and develop in genetics. Genetic diversity is available both within and between the lines or between breeds. So we still have something to work on. Crossbreeding offers multiple combinations, and genomics and robotics are increasing in efficiency. So there are a good lot of elements to help breeding to propose a choice of genotypes for different systems. But then the question remains, at which cost? And now my final message: if you want to have more discussion on poultry topics, you can visit the website of the World Poultry Science Association and join us. And these topics, not exactly the same, but other topics will be discussed this year in the next European Symposium on Poultry Genetics in November in Germany. And there are other working group meetings in 2023, as you can see, in nutrition, welfare, egg and meat, physiology and reproduction, all the calendar is on WPSA.com. There are also meetings organized by WPSA branches in addition to the working group meetings. And then there will be one in France, in UK, in Poland. And the next European appointment for the poultry community is in Valencia next year in June 2024-28 for the 16th European Poultry Conference. So I hope to see most of you there with us. Thank you. Shall I stop the sharing the screen?
Yes. Yeah. Let's see all of us. Okay, thank you. We have a short time for a question or two. If the audience have a question, you can write it down in the chat platform. And I have one of my own. When you compare the percentage of heterozygoty, and you show that the broiler surprisingly have a lot of heterozygosity still. Do you believe that we can keep on selecting for fast growth rate and high meat yield, or the physiology will stop us in a few years?
Yes, I think at the moment there is no real genetic limit because we are still on a relatively short story. If you look at the genetic size, probably of the founder populations and the number of generations, it is not so surprising that we did not get to a plateau. Also because number of traits have been added regularly. So if the breeding goal is changing, then you reshuffle the pressure. So that's why the biological limit, well, they can still be compensated by technology, most of them. It's just for me, the issue of feed restriction of broiler breeder is still something, it's not my specialty, but I still have a hard time to understand how we can improve this. But I think the most difficult limit can be the vision of the society, of the consumer, so more on the ethical viewpoint, the role of the animal, how do we handle the animals, and also the environment. Because the fact that these chickens are producing their heat so much that they are suffering from heat stress already at 20 degrees, for me, I think it is really a shock. And I don't see how we can improve that unless with air conditioning. But then what is the global balance at the environmental and economic level? So I think it's rather, yes, ethical, environmental.
Okay.
And then there is whether
there is one in the chat. Yeah.
Yeah. So
Okay.
then the question of breeding chickens on lower quality diets. Then this is a critical issue because it decreases the dependency on high quality raw material, which often required to suppress forest areas to grow them. But if the growth is slower, the environmental impact is higher because the maintenance cost of the animal gets a relatively higher proportion than the production cost in the use of the feed. So for me, it's really typically a question of optimum. And how to quantify the impact of producing soybean in Brazil to feed chicken in France, and how do we compare this impact with the fact that free-range chickens are running on the ground, but then and you say spoiling some good feed because of running and not of producing broiler meat. So that's why it's really a multidisciplinary approach, and the governance is important because as long as you have a competitor, at least for a breeder, which doesn't care about that, and if this competitor produces a good food at a lower cost, so they take the market. And the nice one, which is thinking, oh, in the future, I should not exhaust such and such resource, but maybe next year they are out of business. although they should be better in the long term. So if there is no governance, it's very difficult to be sustainable, I think.
Okay. Okay, thank you, Michèle. It was a very interesting presentation. I learned a lot, and there was they asked if you can pass the information about the next WPSA conference into the chat box. So if you can do that during the next talk, so they will be able to follow. And we are moving on to the next presentation. I'm pleased to introduce Dr. Yuval Cinnamon. He's an embryologist. He completed his PhD studies at the Hebrew University of Jerusalem. After 2 postdoc fellowships, he managed the genetic research lab at Hadassah Medical Center, focusing on rare human disease. With expertise in bioinformatics, biochemistry, genetics, and genomics, he became a PI at the Agricultural Research Organization, studying chicken embryogenesis and genetic transformation for food security, health, and animal welfare. Go ahead, Yuval.
Good day, everyone, wherever you are in the world. I would like to first thank the EAAP platform for supporting this excellent webinars, and the people from the Poultry Work Group for allowing me to present our research today. And I would like to start to begin with introducing the Golda breed that we've generated in our lab. And the Golda chickens are females which lay eggs from which only females, wild-type females, chicks hatch. And as you can appreciate, this is a rather complex project which involves many aspects, including bioinformatics, tissue culture, genetic engineering, supporting technologies, all the way to regulations. And I will be able to highlight some of these topics during my talk. And I will begin with a simple question, which is probably very obvious to every one of us here in this audience. What do we actually need to Golda? So as we probably all know, 2 distinct breeds exist in the poultry industry: the broilers, which are reared for meat production, and the layers, which lay the table eggs. And together, these 2 breeds contribute to over 40% of dietary protein worldwide. And within the layers industry, unwanted by both the eggs and the meat industries, it is estimated that about 7 billion day-old male chicks are sorted and culled from on the day that they hatch from the females. And this is also possess a huge cost to the industry. It is estimated that each dead male chick costs about $1. So these are big numbers. And with the expected increase in global population, the problem of sorting and culling the chicks is just expected to worsen. And this also generates a major sustainability problems because of the energy which is required to incubate the male eggs, as well as the need to dispose the carcasses. And the US and the EU and other countries in the world are committed to ban the culling of day-old male chicks. In fact, in terms of sheer numbers, the culling of the day-old male chicks is probably the most devastating animal welfare issue in the world. So what are the requirements from a viable solution to this problem? And the list of specific requirements for a good solution that will be adopted by the industry is rather long, and this is a partial list. For example, we don't want to sample the egg. We clearly want the solution not to affect the ability of the layers to lay, etc. But to put it in a nutshell, the solution should be highly accurate, very fast. It must be safe to the layers, and it must be cost effective in order to become a viable solution to be adopted by the industry. And many previous attempts have tried to solve this issue. They focused mainly on attempting to bias the sex ratio between males and females, developing breeds in which the males can be reared as broilers, but almost all these technologies have attempted to generate some kind of a sorting technology to be able to identify between males and females as early as possible during the development. And this concept of sorting, as I see it at least, two fundamental issues. The first is best highlighted in this slide. So as we probably all know, the female hen has just a single gonad, an ovary. And this is a 7.5 days old female embryo scanned using our higher resolution episcopic microscopy. As you can see at this point, it is by the end of the organogenesis, the embryo is almost fully formed, all the organs exist. And if we look at the back of this embryo, we can see that this stage, this embryo, this female embryo, is actually two gonads, identical in size, and this appears identical as males. So the take home from this is that practically until very late stages in development, there are no practical differences between males and females. And the other problem when we are talking about sorting is mistakes, errors. When we are talking about sorting, we're talking about mistakes, and these mistakes cannot be high because this will cause the solution not to be viable. For example, killing wanted females or hatching unwanted males. So what will be the destiny of these males? Will they be reared like they are currently practicing in Germany? This is clearly unsustainable, and this is clearly not benefiting with animal welfare. So despite all these attempts, today there are no viable solution, and this is supported by an article which was published last week from the chairman from the poultry industry in Germany, stating that following the law that bans culling the old male chicks, only few hatcheries will survive. Our solution is fundamentally different, and it relies on two key insights. The first key insight is that practically there are no differences between male and female embryos, at least until too late stages in embryogenesis. And the second insight is that chicken sex is determined by sex chromosomes, and these are the key to our solution. So in chickens, unlike in mammals, the sex determination relies on the Z and W sex chromosomes from the mother hen. If the Z chromosome segregates to the gamete, the embryo will develop into a male, and if the W chromosome segregates to the embryo, it will develop into a female. And it means that the common denominator of all males is the maternal Z chromosome, which segregates from the Z, from the mother hen. So if we introduce a trait, a genetic trait into this chromosome that prevents the development of the embryo, then soon after fertilization, all the male embryos will stop developing, and the females which receive an unmodified W chromosome from the mother and wild type Z chromosome from the father will normally hatch, and they will grow as a normal layers, and they will lay the table eggs. And all this line is by definition containing no GMO material, so they are non-GMO. And this notion was supported by the EFSA organization, and to put it in short, they stated that table eggs, which are produced by the technology that we've developed and can be marketed immediately in all EU member states without any restriction or special labeling. And we are also getting a huge amount of support from the world leading animal welfare organization, Compassion in World Farming. However, the kind of straightforward solution that I just represented has a fundamental challenge within it. So if the male embryo does not develop because it has a genetic trait which prevents its development, how come the female is alive? Because she also has this same Z chromosome, and once upon a time, the female was also an embryo, and so on, all the generations above her. So in order to tackle this challenge, we have designed a unique molecular switch which is activated by an optogenic system, which is basically a genetic molecular switch activated by illumination of blue light, which penetrates through the eggshell. So the trait on the Z chromosome is inactive in all the generations above the parent stalk. And when the eggs of the layers reach to the hatchery, they can be illuminated by blue light, and then the trait is activated. All males stop developing, and only females, wild-type females, hatch and lay the table eggs. So how does this solution integrates within the breeding program of the genetic lines? And I must thank Michèle for her nice introduction to this slide. So there are basically 4 pure lines, A, B, C, and D. And from there, the grandparents are selected, and when they're crossed, they give rise to the parent stock, and then the layers and the males are here, and the layers lay the table eggs. So where do the Z chromosome gets to this Z male embryo? He gets it from his mother, the golden hen, and she gets it from her father at the grandparents from the C position pure line. And only when the egg is delivered to the hatchery at this point, all these steps, generations are alive, and at this stage, the eggs are illuminated, the males do not develop, and the females hatch, grow, and lay normal table eggs. So what are the requirements for the targeting vector to be introduced on the Z chromosome? So there are several requirements for these genetic traits, and we have designed this construct that we call the Holy Grail, and the Holy Grail consists of several elements. First of all, it completes homology arms elements which facilitate the integration of the entire cassette to the Z chromosome. It has a single promoter which drives the expression of the entire cassette. It clearly has the molecular switch, the optogenic system, the lethality inducing gene, and we also had a safe lock element which prevents an unintended activation of the entire cassette. And this is simply a reported gene, green fluorescent protein, GFP, which stops the expression of the rest of the cassette until its removal by genetic means, as I will explain shortly. So where we designed to integrate it, we found a region on the Z chromosome of about 800 base pairs, which showed no RNA transcription. And within this region, we found several sgRNA for CRISPR double-strand break, CRISPR-Cas9 double-strand break, and we scored these sgRNA sequences to achieve or to eliminate or to minimize the chances of to increase the chances to prevent unintended integrations or double-strand breaks within the genome. And we picked one of these sgRNAs, and flanking this region with a small gap between it, we designed 1.5 kilobase homology arms. And then we took the entire Holy Grail, all this construct, and we co-transfected it with CRISPR complex into primordial germ cells, or PGCs in short. PGCs are embryonic cells which give rise in the adult to the gametes. So they differentiate into sperm in males and eggs in females. And we grow these cells in the lab, and following the transfection of these two elements to the PGCs, we Fox sorted them to a single cell according to the GFP, which is a safe lock mechanism, and we let these single cells to form pure colonies which developed, and this allowed us to identify each and every of the pure colonies and to identify a correct integration. And we analyze these colonies using several methods. For example, using solvent blots with molecular probes outside of the Holy Grail, and also a probe within the Holy Grail to allow us to identify off-target integration. And this this kind of solvent blot look like. So we see here a correct band size for integration at the 5' and in the 3' end. All the colonies were positive for the inside, and indeed some of the colonies showed an off-target integration, which was kind of expected, but we were able to identify good colonies. Additionally, we used PCR to the gap between the homology arms, and this allowed us to identify colonies in which homozygote recombination occurred following the double-strand break. So most of the colonies were heterozygote, only one modified Z chromosome, but the colonies labeled with red arrows are actually colonies in which two alleles were targeted. And then we took the cells and we injected them to the bloodstream of surrogate embryos. And when these embryos reached to 10 days of development, we analyzed their gonads and found that many of the PGCs colonized the gonads. And when we let these, not these embryos, of course, but other embryos to hatch, we got these surrogate chimera embryos, and you can see here the window in the eggshell through which the injection was done. And when these surrogate chimeras reached to sexual maturity, we analyzed the sperm from the males and identified chimeras which were positive for GFP, so suggesting that they are going to give rise to founders of the Holy Grail. And indeed, we inseminated these males with wild-type females, and we identified founders of which were transgenic and expressing GFP. Here is a negative leg. I hope you can see that. These are negatives, and this is the Holy Grail first founders. So at this point, we were quite satisfied, and it was time to check whether the construct of the Holy Grail is actually working. So in order to test this, first we had to remove the safe lock mechanism. And in order to do so, we have introduced 2 FRT sites flanking the GFP. And these FRT sites are sequences which are recognized by a recombinase enzyme, which is called FLIP-O. And what FLIP-O is doing, FLIP-O identifies the FRT sites and excises them from the genome. And this will make the promoter driving the optogenes. And so in order to do this, we had to also generate FLIP expressing chickens. And cutting a somewhat long story short, we also obtained FLIPAS expressing chickens. These are also GFP positive. And we cross these chickens with the founders of the Holy Grail. We got all the expected genotypes. Especially, we were interested in this genotype, in this male, which has FLIP-O on one allele and the Holy Grail, and now it is minus the safe lock. It is activated on the other allele. And this is one of the males, and this was his genotype. It has one FLIP, one Z with FLIP allele, and one with the Holy Grail without the safe lock, which was removed by the FLIP. And the next step was to segregate between these 2 alleles. So we crossed this male with wild-type females, hoping to obtain females which only have the Holy Grail minus safe lock. And sure enough, this is Golda. And Golda is a female which, and it is held here by Dr. Inbal Bental Cohen, who is the leading scientist and my dear partner for many years, working days and nights on this project. And the genome in Golda is a promoter. Now the stop is excised, driving the optogenes, and the lethality gene is not expressed because there is a polydylation site which stops the activation of the lethality inducing gene. So what are the optogenes? The optogenes are basically 2 fusion proteins which are bound to inactive halves of an enzyme, of a recombinase enzyme, which is called the Cre recombinase. And the Cre recombinase is an enzyme which identifies LOX-P sites, which are positioned here and here. And when it recognizes them, it excites them also from the genome. And upon blue light illumination, the optogenes dimerize and form an active form of the Cre enzyme. And this identifies the fragment between the 2 LOX-P sites, take it out from the genome, and then the promoter drives the lethality inducing gene. So by blue light illumination, this is the how the genome of the males is supposed to be a promoter, which drives a gene which stops embryogenesis. And what is this gene that stops embryogenesis? And we picked a gene which is by the name of Noggin, and Noggin is a BMP inhibitor. BMP, very important signaling pathway for embryogenesis. Specifically, BMP4 was shown in mice embryos to be necessary for the earliest stages of development, and also in the chicken blastoderm, BMP4, BMP7, and ID2, which is BMP downstream effector, are very highly expressed within the blastoderm. However, Noggin is very weakly expressed in the blastoderm, and only 24 hours later, some expression of noggin can be seen in the embryo. And we hypothesized that if we overexpress noggin in these embryos, we will interfere severely with their initial stages of development and stop embryogenesis. And so we took Golda eggs and we basically illuminated with simple LEDs. And we incubated these eggs for 72 hours, and we identified normal-looking embryos, and these were confirmed to be normal females. And we also been able to identify many severely affected embryos. This is 72 hours after incubation, and it basically, they look like table eggs. You can hardly see anything in these eggs. And specifically, we took this embryo for analysis using the high resolution laparoscopic microscopy. As you can see, it is badly malformed. The brain is deformed completely. The neural tube is open, and if we look at the back of the embryo, we can see many somites, some formation of multiple somites. Normally, they will be just 2 rows of somites in the paramedial area, and there are many somites, and this is with agreement with the overexpression phenotype of noggin. And then we let these illuminated eggs to hatch, and we, out of 208 eggs that we were able to identify that they're males, because with some of these eggs, we were unable to even get a proper DNA for sampling, no males hatched, while in the dark control, which we clearly did, we had both females and males hatched, and females in the blue light, of course, also hatched. And when we compared the hatchability rate within the dark control, it is, as expected, 50-50%, and within the blue light induction, 100% of the hatches were of females. And so we let these females to grow now, and this is an experiment started about 10 weeks ago, and this is a video taken about a week ago, and these females are wild type, and we are waiting for them to start laying table eggs, for which no males were culled within the upcoming week. And with this, lastly, I would like to highlight some of the advantages of this kind of technology. First of all, in terms of reliability, it is a genetic solution. It is expected to be 100% accurate. It is compatible with the current breeding program. It can also maintain the genetic diversity, which is clearly important for the industry. It allows very easy integration to the hatchery workflow. Male embryos cease to develop very early during embryogenesis. Clearly, there is no need to sample the egg like in other solutions. There is no expensive sorting technologies like MRIs or mass specs need to be introduced to hatcheries. It is applicable to variety of chicken strains, types, different color eggs. That male eggs, although containing genetically modified material, they are of high nutritional value, and clearly they eliminate the timely and costly sorting step within hatcheries. It also benefits with the environment and with the sustainability, because using a supporting technology that we're developing, between 48 and 72 hours of incubation, we can exclude the male eggs as well as infertile eggs, and save more than 50% of incubation space, which is required, and clearly there's no need to dispose the dead males. And this is the outcome product is clearly a non-GMO solution. But most importantly, this solution will bring to an end the practice of sorting and culling billion-day-old male chicks worldwide. And I would like to finish with acknowledging my outstanding group of people which have devoted huge amount of time and efforts during the years. I've mentioned Dr. Inbal Bental Cohen, Olga Genin, Noah Semel, Michal Dimur, Tani Kushnir, Izel Cohen, Eva Lamed, Inbar Neubrit, Mike Fan, Amit Haron, Onen Tal, Gabriela Brown, Chagai Vehila. I cannot finish without giving a great credit to my dear collaborator and friend, Dr. Shelley Dorian. Shelley, I thank you so much for everything. You taught me so much on chickens. This project is supported by Poultry by Humane through the Kidum group of the ARO, and I would like to thank you for your attention. Shelley, microphone, please.
Thank you, Yuval. I believe we are not going to have a coffee break, but we will continue. So we have a time for a question or two, if someone has. Let's see. Yeah. How can it be considered non-GMO? Yuval?
How can it be considered
GMO. They are asking, how can it be considered non-GMO? Why have they decided to call it non-GMO chicken?
The Golda is clearly GMO, but the Golda is not the product.
Okay.
The product is the table eggs, and the table eggs are laid by layers that, due to sex chromosomal segregation, they do not contain a genetically modified material by definition. And so they received a wild type W chromosome from the mother hen, and they contain non-GMO, and so is the table eggs that they lay. So this is why EFSA has agreed with us that these table eggs are non-GMO and can be marketed without regulation or need to labeling.
Okay.
Another question.
Okay, I believe so. Another one, and the last one, sorry, we don't have time. Attributability of female exposed to blue light during incubation seems to be lower than that of female exposed to darkness. Yes. In other words, female attributability seems to be impaired with blue light too. How do you explain this effect?
Okay. So we know this, and this is clearly was on the table. We see a decrease in females' hatchability, but we know that this increase in hatchability has nothing to do with the blue light. It is with the conditions that these eggs were exposed, which are suboptimal during the exposure. This is something that we are now bettering. These are the very first results, and we know that these lower rates are not due to the light, but maybe due to somewhat overheating of the LEDs or other ambient conditions during the process, because we have good controls. And we also checked these dead females, and these females die, they don't hatch, but they die at the very late stages of development. unlike the males. So it is not due to being affected by the light.
Okay, so we will stop here. Thank you, Yuval, for your
Thank you very much.
talk, and let's move forward to our next presentation. It is my pleasure to introduce Professor Werner Bessei. Professor Bessei is the Professor Emeritus of the University of Hohenheim, Stuttgart, Germany. He is lecturing behavior and welfare of farm animals with special reference to laying hen, broilers, waterfowl, and turkey. He studied agriculture science at the University of Hohenheim. After his doctorate and habilitation, he was appointed professor for small animal production and farm animal behavior. He served 5 years as the poultry officer at the FAO Rome. Thereafter, he was head of the section Small Animal Production and Behavior. He was a member of various EU expert groups dealing with layers and broiler welfare and participate in various national and international research projects dealing with welfare and behavior. We are so glad you are here with us, Professor Bessei. The floor is yours.
Okay, thank you for your nice introduction. I do hope that the system will not break down. Can you see my presentation?
No.
I think Federico wanted to share it now because we have this problem. I still have it.
Yes,
okay, it's coming now. Okay, yeah. It's coming up now. No, okay, right. You know, I've been working on behavior of poultry and other animals, farm animal species for the last about 50 years, you know. At the early beginning of my work, you know, animal behavior was something of a minor subject among the whole discipline of animal production. But with the increasing interest in animal behavior and animal welfare, so behavioral sciences became more and more important. And now today, you can't talk anymore on poultry production or animal production in general without talking about welfare. You can't talk about welfare without any comment on the behavior. So this minor subject has become, in the meantime, a major subject in that. Okay, may I have the next slide? Federico, okay, thank you. Now, during the last years, a lot of institutions and organizations have been working on welfare of animal production. But among them all, you show that only a few of them, EFSA has in Europe taken the lead in this one. And the whole thing started already in the late 1970s, but what became very important in the 1980s. Next slide, please. Right, so I would like first to give you an overview on the historic development of husbandry systems of laying hens. Why laying hens? Because laying hens were the first domestic animals which have been regulated heavily by national and also EU regulations. And this development, and I also want to focus my talk on the space and group size, because space and group size is like a red line is going through the whole discussion of animal welfare in all species, not only in laying hen. It has started with laying hens, but it's now also very important in all other species, from pigs to cattle to rabbits and what have you. Then I have to draw your attention on this dilemma of space and group size, because the total space to move is really heavily dependent on group size, and group size has special effects on the behavior and welfare itself. We're coming to that later, you know. And the next part will be the change of the human attitude towards animal welfare. I think you can't explain the whole development of the welfare regulations and the husbandry systems without looking back to the human attitude and change in human attitude. So later I'm coming to the conclusions. We have the next slide, please. Right. So it started with in 1970, early 70, almost 90% of our population of commercial layers have been kept in this type of cages at about 4 to 6 birds per cage. And they have got at about 400 square centimeters each. And though the total space was about 1,600 to 2,000 square centimeters. And a lot of these cages are still active in countries outside Europe. Next slide, please. Right. What have been the arguments for the cages? Against the expectation, the birds in under this very barren and limited cages, they showed the highest performance. They had cleaner eggs. They had also better health and even a lower mortality due to aggression and cannibalism. And even long-term experiments in the very early phase of the cages showed that even the longevity of these birds are better than in larger pens. Nevertheless, you saw this cage is really a horrible aspect, for the people who are not used to. And then there were a lot of hassle about these cages. They were not accepted. And but the problem was, that performance at that time was used as a criteria of welfare. So the farmer said, what do you mean, on welfare? Now these birds do have a high production, and without very good welfare, you wouldn't have such a high good production. Of course, this argument is not accepted anymore. And so people had to look for other criteria. And that was the point and the hour when behavior came into the play. So people looked at the behavioral requirement for welfare. And really, there were some deficits in these cages. Lack of space to move, or limited cage space to move. Lack of purchase to rest, lack of substrate to peck and scratch, because scratching and also dust bathing was considered as an important behavior. And of course, also a lack of nests. The hens have a high tendency to look for nest sites to separate themselves from the flock to lay an egg. And so these requirements were then used as the main arguments against the conventional cages. May I have the next slide, please? Right, and those are very important people in Europe at that time. It's almost now 50 years back, that opposed against cages in public, like Konrad Lorenz. He was a very important ethologist, and also he got the Nobel Prize in medicine. There was, for instance, Ruth Harrison, who wrote the book on animal machines. And this book was considered to change the world, the total world of livestock. That means to improve that while opposing against this intensive livestock production. But here she keeps some fur animals, but she also opposed against caged layers. Or finally, also Astrid Lindgren from Sweden. You may know them from the children books. And she also officially opposed against caged hens. And the then Prime Minister of Sweden had to promise Astrid Lindgren to ban cages somewhere. You see, that has had cages or cages layers have had a very high political importance, at least in Northern European countries. May I have the next slide, please? Right. So I will shortly, because I don't have such much time. I was told to talk only not more than 20 minutes. Okay, so we'll have a very short list of milestones, although beginning from the 1980s, there was a lot, really a wave of publications, studies, expert groups, and seminars on poultry welfare. So the first step was really the Council Directive in 1986, which was considered to regulate keeping laying hens and to alleviate some of the most important constraints of battery cages. And that was called, that was a minimum standards, laying down minimum standards for the protection of laying hens kept in battery cages. Please remember the wording. Then the next step was, and this was in this directive, the minimum stocking density or space per bird was restricted to 450 square centimeters. Before it was 400, but in the United States, they were much below this 400 square centimeters of space per bird. The next step was the report on the welfare of laying hens. You know, before we protected the laying hens, now we protect the welfare of laying hens. We will come to that later. And this report was a basis of a decision of the EU Commission to ban conventional cages and to introduce the so-called furnished cages. We will see it in a moment. And later now, there was also still the debate going on because people were still not satisfied with the space which were given in these furnished cages. And then there was a very big study initiated by EFSA in 2005, which were really considering the literature on all these studies and publications which we have had during the last 20, 30 years. But this commission still did not recommend to abandon furnished cages because they recognized that there are some problems in the so-called alternative systems to cages. Problems, especially cannibalism, but also smothering that people and fear and so on. And now the whole situation has got more importance now in just during the last month, you know, because there was a scientific opinion on the welfare of laying hens on farm produced by EFSA. It was adopted in the late 2022 and published just in the first month of 2023. And this scientific opinion stated cages should not be used. That means also furnished cages would not be accepted anymore. Just see the development of wording. First, we had a protection of animals. Then we talked about welfare of laying hens. And then we had the scientific opinion. And that was all stated based on scientific published literature. And now you will see the scientific opinion of experts coming into the play. May I have the next next slide, please? Right. Now, these were the two scientific opinions which came out this year. You may not be informed about that so far because Rust just came out recently, and that was that the cage should not be used. Next slide, please. You can read that. You get it in the internet. The whole files is reports of in between almost 200 pages. You know. Now let's have a look to the development from the 1970s onwards. You know. And you see here you have on the left-hand side, you have the conventional cages with about 400, 450 square centimeters. Then it's coming the furnished cages where the Commission already considered this 450 centimeters as not sufficient for space. So they increased that to 750. And this figures with the asterisks of 890 square centimeters per bird, that's a special German regulation. Right, in this welfare debate, Germany always tries to take the lead, and so to be a little bit better than the rest of the EU. Then we have the aviary system as alternative systems with more than 1,000 square centimeters per bird. And of course, in the free range, you can add 2.5 square meters. So you are coming to this 26,000 square centimeters per bird. But what you see in the next line here, that we have had only 4 to 6 birds per cage in the old system, the conventional cages. This was increased in the furnished cages to 10 to 60, sometimes 90, but more birds. But this was because we could not accommodate all the equipment. That means the furnished cages, which have perches, nests, and dust baths, could not be accommodated in very small, these 4 to 5 bird cages. We have had that in the early beginning of the development. For instance, the Edinburgh modified cage was a 4-bird cage with all this equipment, but this was not practical. I could not transformed into a commercial system like these furnished cages. Then we are going to the barn system or aviary system. The group size is going to several thousands, is almost unlimited, or in the same as also in the free range, although in Germany, the group size in barn system, aviary system is limited to 6,000 per group. And you see the total space to move, which is extremely important in the discussion on welfare, that was increased mainly through the increase of group size. So you are coming in the conventional case in between 1,600 and 2,700, 3,000 square centimeters total space, which was really restricted. Then if you go then to the furnished cages, you are coming up to at about, depending on where you have the EU recommendation or the German one, you're coming between 7,500 and 53,000 square centimeters. But then if you go into barn system, that's a total different dimension. We have that in square meters between 600 and more than 2,000 square meters. And of course, in the space to move in the free range is going to hectares if you're going to several thousands of hens per group. And here you see down the old cage, and then they have this furnished cage with this equipment and the aviary, and on the right-hand side, the free range. Right, and so next slide, please. Right, okay, the first approach to measure the minimum space for hens, you see that is a publication of Marion Dawkins and Hardy in the end of the 80s, just to measure the space covered by the bird at different behaviors, which is right, the most space is being occupied when the bird starts wing flapping. You see it on the right-hand side, which is almost 2,000 square centimeters. And so, but not all of birds are wing flapping at the same time. So I think we cannot take the maximum space for one behavior as the maximum space in average of the whole thing. Next slide, please. Now, in the new opinion on welfare, they have had an extended approach to measure this minimum space required. They modeled the hens as a circle, and then the core side in yellow, that is the hen, but they added a certain area for social space. And in addition, they added on the right-hand side, this space in beats birds is another 10%. So in total, using this equations to measure or to lay down the minimum space, they're coming to more than 3,000 square centimeters. Next slide, please. But still, this was not enough. You see that, oh, can you skip that? I think you jump over that. I think otherwise we're getting, it's getting too short. Next slide, please. Yeah. All right. Now, how can we now explain the jump from the previous report on welfare on the new opinion of EFSA, of the working group of EFSA on the new decision not or to allow cages anymore. Right. And now we have to consider that there was also a change of the methodology, the whole thing. So this decision was made on a so-called EKE or EKE methodology. What's the expert knowledge elicitation? Now, what is that? Now, this expert knowledge elicitation is a method where the experts are asked, what do you think is a minimum space a hen could have without any restriction, a minimum space without any restriction of the behavior? What's the minimum space where there's no limitation? Okay, if you have fixed that figure, then you are asked in the second step, how certain are you? Are your certainty 100% or the 0% or around at about 50%? That is roughly the idea behind the thing. Anyhow, this EKE presents more or less this, a subjective figure. Of course, it is based on the knowledge of the experts in poultry science, right? So, and they came to the, and that's important now, this jump, they came to the conclusion that the minimum space to move in the depends on the group size, and they made a split between group sizes up to 30 birds, though the minimum should be 25 square meters. That means if you keep 1 or 2 hens, they should also have 25 square meters. Of course, when you have more hens, they can share this space. And when you have more than 30 birds, the minimum space should be 80 square centimeters. That can also be shared by the different hens, And there was another figure elaborated on the space, that we should not have more than 4 birds per square meter to fulfill the requirement for comfort behavior and to reduce feather picking. That will be euphemistic because we know that feather picking does not heavily depend on on stocking density. It rather depends on group size, what we know. But anyhow, that was the argument of the expert group who developed these figures here. Right? I will not talk about the pullets, and this is also applied to pullets, but we go further on laying hens. May I have the next slide, please? Right, the dilemma, we are coming now to the dilemma, just in the case of cannibalism. We know the detrimental effect of group size since ages, even before these small cages with 4 to 6 birds were developed. In the early phase of this caging, we had colony groups of more than 20 birds, 20, 60 birds, as we have now in the furnished cages, And we knew that we had problem with cannibalism. We had higher mortality, we had aggression, and we had a high flightiness sometimes, which developed what was called at that time hysteria. That means the birds are getting really excited, the groups, and they fly to one part of the cage or the pen and pile up over there, and the birds on the lower level will be suffocated. So we have, through these outbreaks of hysteria, we have had a high mortality sometimes in these groups. And this we know that was a reason why these colony cages became smaller and smaller until we ended up with about 4 to 6 birds per cage. That was before we had these so-called conventional cages. The same problem of larger groups was also became clear when people developed also in the 70s and 80s the so-called welfare cages or getaway cages. It was started in England, came down then to the Netherlands, to Switzerland, to Germany, when we tried to develop larger cages with pens, with perches, with nest sites, and with scratching mats, or with really sand baths, And that also failed. We could not develop that, came not to a commercial level. And at that time, I just found a citation of the expert group of the EU, not EFSA at that time. They stated, generally speaking, all papers dealing with group size showed that increased group size creates problems. Right? You see the dilemma. Either you have more space to move, and then you're running into other problems, or you have the small groups, then of course it will be a problem of movement, to exhibit their natural behavior. All right, may I have now the next slide, please? Yeah, right. So the early furnished cage, which you see on the right-hand side, this was a first furnished cage which was developed. No, not the first developed on the commercial cage. There was another one in Sweden, but we also have had it in our research station that was constructed for about 10, 12 birds, because we thought we should give the birds perches. You see the perches, you don't see the dust bath in the back, but we have a nest site on the right, which is separated from the rest by these plastic flaps. And also you have a nest floor on plastic mats, which is not a wire because it was a requirement. The nest site should not be covered with wire floor. So, but this was to our meaning at that time, a trade-off of the detrimental effect of group size of large groups and of the requirements, the basic behavior requirements for nests, perches, and scratching areas. I think this now the change or the idea or proposal to ban this furnished cages now are difficult to understand, right? Because we considered that, even the EFSA considered that as this compromise between behavioral requirements and the potential damages. And of course, larger groups, as I said before, we have also the risk of cannibalism. Right? So we cannot explain this change of the attitude. We cannot explain this by the change in the behavioral requirement of the hens. That has not changed a lot. A laying hen from the behavioral status of the 1970s to the 2020s is really the same. It's different broilers, but the laying hens is almost the same. I think, Shelley, you would agree to that. Okay, may I have the next slide, please? Right, as I told you before, I consider this change in the regulation recommendations for laying hens, it's rather a change in the human attitude to poultry and animal welfare in general. And you see that in the also expressions, First, I told you we started with the protection of animals. That means we protect them against the predators, but also we gave them the right for basic requirements. That means feed, water, and also shelter. That means a protection against adverse climatic conditions, cold and heat. That was in the early phase of the development. That was they were the main criteria for welfare. Now we moved to welfare, and at that time it was protection of animals. Then it became welfare. We protect the welfare, and there was now an interesting jump in the definition of welfare, and there was a so-called 5 freedoms. I don't know whether you have heard about that, but this came from the Farm Animal Welfare Council in the United States, in England, right? And they reformulated the whole thing. And then that means supply of feed and water was now named freedom from hunger and thirst. That means we don't have to feed and water the birds, but while doing so, we elicited positive emotions. So there was a shift from the real world into the emotional world of humans and also of the birds. And so we had also to free the birds from diseases, from fear and distress, and they should have the freedom to express normal behavior. And at that time, also in the 1990s, there was one more aspect coming into the play. It was the presence of positive feelings. That means in the upper part of the thing, we tried to prevent or protect the birds from adverse feelings and adverse experiences. And now we added that complete welfare of the birds would also comprise the presence of positive feelings. And now we're coming to the so-called enrichment of the environment. Because of the shortage of time, I didn't come, I won't go into these details because this would require another half an hour or an hour. But this is now another development in the whole thing. In all husbandry systems, we not also have them to give them the environmental condition for which are required for production, reproduction, maintenance, and survival. We also have to provide the birds positive feelings like in the human society. May I have the next? Right. So there was a big and long time EU project, EFSA project, which developed the so-called welfare quality protocol in detail, where we have, besides the basic requirement, physical requirements on the space and so on, we have also a lot of tests to appraise some fear, for instance, or gait. So the assessors, they went into stable and made these tests, which I would say are semi-subjective. These tests are standardized, the score system are standardized, both the gait score, but also the fear score, the approach to human test. But still, there's some subjective components, and then that means you cannot be free of subjective influences when doing these tests. And also, it is really a little bit difficult to go to a farm which you don't know, the flock doesn't know you, and go and within a short time you appraise the total welfare. But this subjectivity is going on. May I have the next slide, please? Yeah, that is a little more in detail the principles of or the system of the welfare quality scoring system. You have 4 principles: that good feeding, good housing, good health, and appropriate behavior. And then you have the different criteria how to assess it, and then you have to examine how to measure it. Of course, for good feeding, feeder space or drinker space, which is easy to do, but it's getting more and more difficult if you go to good health, but it is still possible. And the absence of pain induced by management procedures, big trimming, of course, you can see whether the birds have been big trimmed or not. And then you have the expression of social behaviors. That's the expression, plumage, plumage conditions, or comps and wants mainly on the comp of the bird. And then the use of nest, litter, and so on. So, and you have seen this avoidance or fear test of human beings in the previous slide, and you see you can also see how the birds respond to humans, although in this case the human is not familiar with the flock. But the most and fear, the fear test is also with novel object test is also good standard to standardize. But now we are coming to the most on the right lower level. Here we are coming to the so-called qualitative behavior assessment, or QBA. What is that? Right. But before we are coming to that in more detail, I just want to show you some critical points of this whole system. First of all, in this, you're making then a general welfare score of all these items. But in the system does not allow trade-offs between the principles. Just one example. If you go, it was Michèle told us that the special broilers are very sensitive, more and more sensitive to heat when they're growing up, right? So in the conditions of Brazil, where it's very hot, they automatically run much lower. They can only support much lower stocking densities than in other countries. Also in Israel, they have lower stocking densities if the stable is not climatized. And this space, this stocking density, is getting very high scores. If you have low stocking densities, you're getting a lot of benefits on the score. But you cannot compensate that by good ventilation in the scoring system. This, the mathematical construct of the score prevents this trade-off and this balance between the scores of the different principles. And that's one thing. And the other one is that the QBA, what you see, that is extremely based on the feelings of the observer. May I have the next slide, please? Here you have a list of the positive and negative feelings which the observer is supposed to score. So whether the birds are active, relaxed, comfortable, fearful, right? Fearful, you may see, you know, rated, whether it's agitated, but now it's confident. It's depressed. How you state a depressed hen? Calm, it's okay, but then friendly and happy. What is a happy bird? Happiness is maybe it's when they start dust bathing or wing flapping or running and so on. That's a happy what we think. Or we have negative feelings like bored, do they look bored, tense, unsure, and so on, frustrated, scared, nervous, and distressed. These are really subjective feelings. And I skipped some slides where I showed you the problem. You know, if you want to make a conclusion from our feeling to the feeling of these are very human approaches, you know. This reflect human feelings, you know. And if you want to make a conclude from human feelings to animals, you know, you have several thresholds. First of all, the environmental condition, which is perceived through the sensory system of the bird. Then it's processed in the central nervous system, and then is produced the emotion on that. So you have 3 different levels, and you compare these 3 different levels of animals with humans. So, and I think these thresholds are really difficult, but on each level you have differences of the, or different processing or processes. For instance, pain can be transformed on the central nervous system from a high excitement, can be damped down to almost nothing, depending on the situation, right? So it is really difficult to do this conclusion and to bring it to a scientific level. And that is my main criticism, that it's really difficult. But of course, we all do that, you know. We all rate the feelings of our dog, maybe of our cat or dog, or the animals, horses, and so on. We do it. But to bring it on a scientific level, it's almost at the time being not possible, unless we are going to the so-called functional magnetic resonance imaging, which on the brain of the birds, which is being done in the human beings, which can where you can show whether certain centers in the central nervous, which reflect positive or negative emotions, are active. If we could transfer that to our animals, we could really verify whether this criteria was on the welfare protocol, this QBAs, could be realistic or not. But so far we can't do that. Next slide, please. So yeah, sorry, I didn't see that it was here. You know, I have got this again. This approach of the threshold we have to overcome, where with the sensory system, the central nervous system, the behavior of the birds, and here we have the observer, and now we have in each stage, you know, we have to make the analogy between animals and ourselves. And so there exist uncertainties on all levels which cannot be overcome through the sophisticated statistics as we use and apply in the EKEY, in the expert knowledge elicitation system. May I have the next one, please? Yeah, that's the fMRI, which are applied now in human beings, and there are nice models. And it also works in rats, and we have nice models to explain how we can really rate or establish the emotional state of an animal by applying this system. But it's not, we are far from having the technique to use that in farm animals in their normal environment. In the lab, may be possible. Next slide, please. Right, in the conclusion, the development of welfare regulation was driven by a change of the human attitude and or opinion on animal welfare rather than on the change in the requirement, behavior requirements, in the physical requirement of the animals. And in analogy to the development of welfare of the industrial population in Europe, the requirement of welfare increased from basic requirement needs, food, water, protection from extreme environmental conditions, safe housing, freedom from discomfort and provision of comfort, and the freedom to move has become the most important, more important than the related risks of cannibalism, like also smothering, this crowding, bone breakage, diseases, and predators. And coming back to the talk of Michèle, I think this freedom to move has also overcome all economic aspects of that. Economics are a no-go anymore in the discussion on animal welfare. Economical results were still considered in the earlier reports on the behavior and welfare of laying hens and broilers, but that has been faded out totally as a no-go. And therefore, we are coming to this point that now we are going to have more free range. We're going towards more free systems. And interestingly, this freedom, it has become a catchword. And therefore, it was so successful. You cannot sell, if you go to marketing, the market, you cannot sell scientific results, but it's very easy to sell emotions. And in the United States, this anti-cage activities of NGOs, they are arguing not with aviary or free-range system. Those they say, no, we are having non-cage system because the cage is in the public meaning is the worst you can do to the bird. So freedom, cage-free, that's a really slogan. And all other aspects, even environmental aspects, as Michèle mentioned, this complex of all aspects are really subordinated to this idea of freedom in the animal welfare. It's an unfortunate development in the welfare debate, but that's a fact. Now, where are we going now? May I have the next slide, please? Right, what's going next? Now, ban of aviaries. If you follow the line, the whole line, I think even the aviaries will not be accepted in the future. And interestingly, in Germany, we had a time at about 1 year when we did ban the furnished cages before the EU came to this idea. That was already 20 years ago. At the early beginning of the furnished cages, it was banned by one government for a while. And already when the furnished cages were banned in Germany, then the NGOs started to argue against aviaries. They didn't like aviaries. And I think, to my opinion, for the welfare of the hen, a small furnished cage is in total better than a large aviary. But anyhow, they already started and they know the disadvantages of large groups and aviaries. But then the furnished cages were allowed again. There was a change in the government, and then the NGOs stopped the lobbying against aviaries because as long as we have furnished cages, we have to go to argue against furnished cages. What's again next? And I think at the end, in the EU, only free-range systems will be in the future will be accepted. So the ban of pig trimming and other treatment, really ban of pig trimming is always discussed, and I think in the long run it will not be allowed anymore. But that raises the problem of cannibalism. If you don't pig trim, you will have cannibalism. And there was a saying of a colleague of mine from the Netherlands who was totally against cages. But he told people always in all the seminars, if you want to ban cages, you have to permit pig trimming. Now we have both. We banned conventional cages. There will be a ban of furnished cages, but we still allow pig trimming. That will also be banned. Anyhow, then ban of killing the old chicks and layer lines. We have heard about that. That is already established in Germany. But I was really glad to hear your talk, Yuval, because when we started this debate on killing the old chicks, I told people that the solution will be on the genomic manipulation. But this is not allowed in Germany anymore. We couldn't do these experiments as you did in Israel, or can be done in Asia or in the United States. Anyhow, there was no go in Germany to follow this pathway, and I'm really glad that you succeeded. The other one is a ban of forced molt. That is also an object. I think forced molt is a wrong wording, because forced molt used to be induced by withdrawal of feed, water, and light. It's not allowed anymore, but there are more human, I would say, human methods for a laying break instead of forced molt, which could be welfare friendly. And then ban of feed restriction, that is applies to mainly these broiler parent stocks. I think that will be, and again, Michèle has told us that will be a problem, but we don't have a solution so far. Okay, that is all what I tried to tell you. So I'm thank you very much for your attention.
Thank you
I'm
very
just
much.
looking for your questions.
Okay, so I have one. Michèle already answered for a part of it, but we would gladly hear your opinion also. How does this approach combine with the sustainability? You need more floor area per bird, and performance are lower due to higher mortality, higher feed consumption, and increased floor dirty and broken eggs.
We have had this discussion in Germany, and clearly even our Green Minister for Agriculture, he declared, no, welfare is rating higher than anything else, even the environmental issues. Quite clear. But you are right, that is a problem, and we can't have both. Now they try to tell us, you okay in this free system, non-cage system, you can control everything, but you can't. It's not realistic to control the environmental issues, because that depends on feed intake and feed conversion, which is definitely worse in the non-cage system than the cage systems. But as I told you, I do not know the opinion of EFSA. But what I try to show you now, that EFSA is really focusing now in the last reports, is going more and more and focus on welfare only. Okay. At least this group was not allowed to, and even in the working groups I attended many years ago, EFSA tried at that time, EU, the Commission, tried to put some influence on the working group not to include economical issues. Okay. This will lift up to somebody else. I don't know how they're going to treat at the end. And it's also not decided in the EU that furnished cages will be banned now. But usually after such a strong statement of a working group, the EU is going, especially in this situation now, and the high pressure from NGOs, they're going in this direction. Okay. But all other you don't believe it, Michèle, but all other things are going to be subordinated to the whole aspect of welfare.
No, okay. I don't say I do not believe it. I just said that it is going into the wall that we will have only pets and no livestock in Europe any longer if we continue on this philosophy. But that's the main threat that goes together with veganism and invite for meat. That because we want animals to be as happy as us, or maybe more happy than people, then we manage a way to eat something else. The main issue is how is it in the other parts of the world, because Europe is only Europe. And that's the big issue for the breeders, because as I said, they have to propose their genotypes for all over the world, and the concerns are different. And I don't know if you remember the comment from Edir da Silva from Brazil in a conference in, I think it was in 2016. This was very diagnostic of the situation, because in Brazil, they are producing a lot of chickens to sell to Europe.
So, yeah.
And you remember his question? He was really honest in his question when he asked you, but Werner, all this story of welfare, this is just to put economic barriers for our exportation. I think he could not see that maybe it was not from an economic viewpoint, that it was just a subjective or a cultural value. Himself, he said, okay, you are just using welfare to prevent us to sell chicken meat to you.
Right.
No, you remember this was really
yes, I know this situation. And look, but that's our government, at least in Germany. But with regard to killing day-old chicks of layer lines, France is going to follow, as I heard. You know.
Yeah, but I think this
Yeah.
issue is I say it's difficult to continue like that. It's different,
but
different from the way of raising chickens. If you just want to have 2 chickens per person in the country, then yes, we will have pet chickens, and we will go back to the 50s. Or but it's all a vision of the social model. But in Europe, it's going this way. My concern is that we are not the biggest part of the world in Europe. So what about the rest of the world?
We are following you.
No, they will not follow. Oh, not that fast.
Not that fast.
Not that fast. Not at all. You know, but you know, our government, especially the German government, is really ruthless in this regard. You know, Yuval showed the comment of these hatchery manager. You know, that most of the hatcheries now, especially the small hatcheries, are going down the drain in Germany for this reason. You know, but they don't. All right. So what, you know? So they're going to Spain or somewhere else, you know, where this is allowed. And of course, that's a way. But you know, at the time being, maybe there will be a change again in mind. But at the time being, is going this direction, that we are going just to transfer the primary production. That means production of broilers and eggs to other countries which have less strict welfare regulations. And we will have a certain amount of birds, layers, and broilers on a small level, which are welfare friendly for those people who are prepared to pay for.
No, but I think this is a consequence also of extremely 100% economic driven objectives for so many years. And then all of a sudden we are on the other side of the moon. And this is typical in social. You remember there was an European action in 2002, and they invited a sociologist. Those people who do not meet so often in our professional life. And I always remember what he said: the faster you go in one direction for one motivation, the faster the reaction will be in the opposite direction. So the fact that we have these big chickens who are refrained from eating for most of their life, I find this shocking. And we are harvesting the consequence of what has been done in one direction. If we had thought of that a bit before, then we could avoid maybe extreme reaction, because if we have extreme in one direction, then we have opposition extreme in the other direction.
Yeah. Okay.
Right, it just depends on the time, you know, because you know, the whole cycle lasted about 50 years from the early free range to the intensive that we are coming back through the cages to the free range again, you know. Right, could be.
Okay, about time. We are 25 minutes late, so I wish to thank you all I hope you enjoyed the webinar as I did. I learned a lot of new stuff today. Amelia, would you like to wrap up quickly or just to invite everybody?
Yeah, just quickly to invite everybody to the next EAP in Lyon. We will have 2 sessions, one in how can poultry farming systems evolve to meet the major societal and environmental challenges, and another one, the PP law session. And if you want to participate, choosing new themes for the next year, for the next EAP next year, then you have to attend the P Commission meeting on the 31st of August. Yeah, so we hope to see you there. I'm also very thankful to the 3 speakers we have. I really enjoy the presentations. I really enjoyed this chatting at the end. And I wish you all a very good end of day for those that are in European times. Yeah.
Thank
Okay.
you.
Thank you very
Thank you,
much.
Amelia.
Thank you all.
We'll see
Bye
you.
bye.
Bye.
Ciao.